Welcome to the Bio 341 lecture on evolution at multiple loci. In this lecture, we're going to cover the concept of genetic linkage, recombination, linkage disequilibrium, epistasis, and finally hitchhiking and genetic draft. So, recall that there was a debate between the Darwinists and the mutationists, where the evolutionary biologists, the Darwinists, were looking mostly at continuous traits that vary in a normal distribution. And the mutationists or geneticists who were studying discrete traits. And recall that Darwin's genetic mechanism didn't work. He had this idea about gemmules. They were supposed to mix between the two parental strains and create a kind of a blend between the two parental strains. But that just doesn't work as far as what we observe in nature, where we typically get out our parental strains in the F2 cross. Also recall that we do know that when we have variation at multiple loci, this can result in a trait that seems to be continuous because each phenotype is a slight gradation from another phenotype. Now, the geneticists also had some problems with their models. We've all done a Punnett square that predicts the classic 9:3 to 3:1 ratio of various traits in an F2 cross. Well, William Bateson, Edith Saunders, and Reginald Punnett actually were working with the same pea plants that Mendel had used in 1905. And they did this cross between pure breeding strain that had purple flowers and long pollen and a pure breeding strain that had red flowers and round pollen. And they crossed them together to create F1 hybrids, and then they allowed them to create F2 hybrids. And when they looked at the F2 hybrids, they found that the ratio of genotypes was not the 9:3 to 3:1 that they expected. Instead, they had excessively high numbers of individuals with purple flowers and long pollen, and excessively high numbers of individuals with red flowers and round pollen. So these two traits were sort of seeming to stick together. And from this, they devised the hypothesis that the two genes were somehow linked. And today, now that we understand the structure of DNA, we know that traits can indeed be linked because they are on the same chromosome and close to each other. This is what we call physical linkage. The easiest example to demonstrate physical linkage is in a system we've already encountered in bacteria. We can imagine during the evolution of antibiotic resistance that we have sensitive strains and we have resistant strains. And the sensitive strain does fine when there's no antibiotic, but does very poorly when antibiotic is in use. Now we have a mutation to one locus that provides resistance to the antibiotic. This strain does well in antibiotic and equally well when there's no antibiotic in use, but it still doesn't do as well as the original strain. So it's suffering a trade-off in its growth and its fitness in order to be able to deal with the antibiotic. But then there's a second mutation that occurs, a compensatory mutation, that then allows the mutant strain to do equally well as the wild type when exposed to antibiotic. We saw an example of this in Baim et al., where there was, while there were some bacterial strains of some of those E. coli that would have a single mutation that would bring them to full fitness and able to deal with the antibiotic, many of the strains, shown here in purple, had to first have one mutation that gave them resistance to the antibiotic, but traded off growth rate, and then a second mutation which allowed them to have a higher growth rate, this compensatory mutation. So those two genes in bacteria are inextricably linked because the bacteria have a haploid genome. we eukaryotes have a diploid genome, and during meiosis, the parental chromosomes recombine. So the maternal and paternal chromosomes will cross over during prophase 1 of meiosis and exchange some genetic material. By mixing up the loci this way, we're able to create new variants. So when locus A and B are on separate chromosomes, it's possible to create gametes that have every single combination of big A and little b inside them. When they're linked and on the same chromosome and close to one another, then it's only possible to create gametes that have the big A, big B, and little a, little b. When you see an association of two alleles that is greater than what you would expect at random, it is likely due to linkage. Because loci that are close to each other are less likely to recombine, and loci that are far apart from each other are more likely to recombine, it's possible to create what's called a linkage map of a chromosome without actually ever looking at the chromosome itself. An inquisitive Columbia University undergraduate in Thomas Hunt Morgan's lab decided to analyze these frequencies of linkage, these pairs of co-inherited traits. So one night when he was supposed to be doing homework, he was an undergrad at the time, instead he sat up all night and looked through the Thomas Hunt Morgan lab's data and created this first linkage map of these 5 genetic traits, inferring, for example, that locus B and C were close to each other and P and R were close to each other, and that B and C were distant from P and R. And he even named a distance, the distance along the chromosome, after his advisor, Thomas Hunt Morgan. He called it a centimorgan. So a centimorgan is a 1% chance of recombination between loci per generation. So the 1.5% chance of recombination between B and C, and a 30.7% chance of recombination between B and P. And recombination is what allows us to explore the full range of phenotypic variation. If all of our genes remained linked together, we would only be able to explore within very small realms of variation. When a mutation occurred at C, for example, we'd be able to explore just within this realm of variation. By allowing recombination, a mutation that occurs anywhere in the population through sex can be recombined with any other allele at another locus, so that all possible combinations at all possible loci are created. This is what allows us to access what's called latent variation in the population. So one piece of vocabulary for you: a haplotype is a haploid genotype. So it's the set of alleles that are carried on a single chromosome. In this example here, big A, big B, big C would be a haplotype that would be combined with another haplotype like little a, little b, big C. Those two haplotypes would form the diploid chromosome of an individual. So when recombination leads to random segregation of alleles in the population with respect to the two loci, we get linkage equilibrium, meaning that they're effectively unlinked from one another. This occurs when at locus B, the big B allele is found on big A chromosomes just as often as it is found on little a chromosomes. And as a proportion, that actually turns out to be true. So if we count the number of big B on big A chromosomes, that's 12 out of a total of 15 big A chromosomes, that's 0.8. Big B is found on a total of 8 out of 10 total little a chromosomes, and that is also 0.8. Because these two proportions are the same, this tells us that the two traits are essentially segregating randomly, and the traits are therefore unlinked in linkage equilibrium. When we don't see a random occurrence of A and B together, then we have linkage disequilibrium. So in this case, B, big B, is found on 11 out of 15 big A chromosomes, but big B is found on 9 out of 10 of little a chromosomes. So big B is associating with little a more frequently than it's associating with big A. And then we say that there is linkage between the A and B loci. Another way to do this that is perhaps a little bit conceptually easier is to is to get the allele frequency for big A and get the allele frequency for big B. So there's a total of 25 chromosomes here, and big A occurs on 15 of them. So the big A frequency is 0.6. There's big B occurs on 20 out of 25 of these chromosomes. So its frequency is 0.8. And then we find the frequency of the big A, big B haplotype. So that's this haplotype, big A occurring with big B on a chromosome. And we find the frequency of that, and it's 12 out of 25, or 0.48. So the haplotype frequency is the same as the allele frequencies when multiplied together. This tells us that they're occurring with each other more or less at random. We can do the same calculation for populations that's at linkage disequilibrium, and we find the proportion of haplotypes that have big A, big B to be 11 out of 25, 0.44, but the proportion of big A and big B is 0.6 and 0.8. When you multiply those together, these are not equal to each other, and therefore these two loci are potentially linked. And this leads to a mathematical definition of linkage disequilibrium, as a matter of fact. It's the difference between what we expect. If they're randomly segregating, then we would expect the proportion of chromosomes that have a big A and a big B to be the product of the frequencies of big A and big B. We subtract that from the actual observed haplotype frequency of big A, big B, and that gives us D. And D ranges from negative 0.25 to 0.25. It's important to note that you only get new allelic combinations when there is recombination in a double heterozygote. If you have a single heterozygote, so this individual is homozygous at allele A, at locus A, and heterozygous at locus B, if you have a recombination between these two chromosomes, you still end up with big A, little b, big A, big B, the same as the parent. It's when you have double heterozygotes that you're actually able to create new combinations. So we start with one haplotype of big A, big B, the other haplotype, little a, little b. Recombination gives us big A, little b, and little a, big B. And so this underscores the importance of having genetic diversity in a population and having as many individuals be heterozygous at as many different loci as possible in order to explore all of the latent variation that's out there. So, processes that create linkage disequilibrium. The first and most obvious is just physical linkage on a chromosome. So if two loci are close to each other on a chromosome, then they are linked. And they will not recombine that often, and so there will be in linkage disequilibrium with each other. That means they will not occur, the allele frequencies will not occur randomly with respect to one another. Other processes that can create linkage disequilibrium, though, are natural selection and migration. So this just shows that linkage disequilibrium decreases as you get two loci that are more and more distant from each other. So each dot here is a pair of loci where we're measuring linkage disequilibrium, and you can see that loci that are very close together have fairly high linkage disequilibrium. Loci that are far apart have relatively low linkage disequilibrium. So it's just a function of distance along a chromosome. That should make sense. However, it's also possible that two loci that are on different chromosomes can be in linkage disequilibrium with one another. And this occurs because they are linked to one another through some developmental process. They are used together in some pathway, and whatever occurs to one has an effect on the other one. When several genes are linked this way in a cellular process, we call this epistasis. So a simple example of epistasis would be a pathway where we have one locus that provides white paint, another locus that dyes the white paint blue, and a third locus that draws a circle with the paint. We could imagine an alternate allele where one locus provides white paint, an alternate allele dyes it red, and the third allele paints a red circle. If one of these loci breaks, then the whole process is broken. So if locus A breaks, then there's no paint to add color to and no circle to draw. And it breaks the entire process. So selection that occurs at locus A will affect locus B and locus C. So an example of this would be two different pathways that create the same molecular product. And if you have a working dominant big A locus, then you don't need the B locus because you're able to create the product. Also, if you have a working B locus, then you don't need a working A locus. So individuals will be healthy if they have a big A or a big B, which means all of these gray genotypes will be healthy genotypes, except for individuals that have the little a, little b haplotype. Those individuals will not be able to make the molecular product and therefore will have a genetic disease. A real-world example of epistasis would be in agouti mice. Agouti is this kind of brownish speckled coat color that occurs as a dominant gene, big A, at the A locus here. And the recessive gene, little a, produces this solid black color. However, if the mouse is homozygous recessive for the C allele, little c, then it doesn't create any pigment at all, and it's not able to create either the agouti or the recessive black coloration. So locus A is epistatic to locus C. Selection that occurs on locus C will affect the outputs of locus A. And finally, migration can have an effect on linkage disequilibrium. So if you have one population of lizards here that is fixed for big A, big B on its chromosome, big A, big B haplotype, and you have another population of lizards that is fixed for little a, little b, if you have migration from the mainland to the island, and you bring in this little a, little b haplotype, the descendants of this individual will tend to have little a, little b in their genotype, and little a and little b will stay associated with each other for some time after the migration event. However, recombination will eventually break them down. So remember, recombination is what breaks down linkage disequilibrium by mixing up allelic combinations. And over time, linkage disequilibrium will break down. So this is a measure of linkage disequilibrium, and this is now time on the x-axis rather than distance along a chromosome. And you can see with a low recombination rate, no disequilibrium doesn't break down. But with relatively high recombination rate, we can get a breakdown in linkage disequilibrium. And therefore, alleles on different loci will associate randomly with each other. So the recombination rate, we can look across of a variety of different species, and we see that the recombination rate varies fairly significantly. But when you look at the recombination event per chromosome, it always ends up being right around one recombination event per chromosome per generation. And that makes sense. There's only one crossing over event that occurs per chromosome per generation. Now, if you have really strong selection, such that it brings one locus, one allele at one locus to fixation, because it's a highly beneficial allele, we call this a selective sweep. And a selective sweep will tend to drag the other loci, the alleles at the other loci that are nearby this beneficial allele, it'll drag those to fixation as well. So because a big B allele at the B locus here is beneficial, there will be a lot more copies of big A, big C, big D, and big E in the next generation, not because they're beneficial, but simply because they kind of came along for the ride. And we actually call this hitchhiking. These loci here are not beneficial, but because they're linked to locus B, they're brought to fixation. And so you can imagine several different haplotypes, each of these haplotypes showing genetic variation. And you have a strongly selected beneficial mutation that arises on this particular haplotype. Most of the haplotypes in the next generation will have the genetic variation that was present at this haplotype and which are nearby this locus. And so it'll decrease genetic diversity in the next population because all of these loci will come to fixation. So again, this is hitchhiking. And we can see evidence of hitchhiking in the lactase allele. So we've talked about this before, how lactase is an allele that is expressed in the wild-type human, is only expressed in infants and not expressed in adults. However, lactase is expressed in adults of cultures that have domesticated cows and produce milk, because it is advantageous for adults in those populations to be able to drink the milk. So if we look on the x-axis, this is the location of the lactase gene, and this is the distance away from that gene on the x-axis. And these are wild-type humans that do not have the adult expression of lactase. And you see that there's no evidence of hitchhiking. In red are these very distinct bands of highly similar haplotypes that have occurred in European and African populations that have domesticated cows. And so this is evidence that these other loci are getting brought to fixation by selection on this lactase allele. Another word that we use instead of hitchhiking is kind of a play on words on genetic drift, but we call it genetic draft. So these loci are drafting off of lactase allele to become successful, even though they themselves are not selectively beneficial. So these are the concepts that I would like for you to understand from this lecture. And there's really only one learning outcome associated with this.